So, here we have an arcuate vein, an arcuate artery, an interlobular artery, an interlobular vein. Okay. What they've done, the colors that they've used in this model are green for the capsules, you see? They made green capsules. Fine. They haven't sliced them. I would have liked this model to have at least one of them sort of sliced to show you a glomerulus inside. They didn't do it. Well, that's their company. So here we have, from here, going to the capsules, these are what? What is this? An afferent arteriole. And what are these? Efferent arterioles. Okay? Now, here, let's look at a nephron, okay? We ready to look at nephrons? All right? We're ready to look at, you want to look at a nephron? Could you see over here? Or you, okay. Now, in beige, I guess you call it beige? Mm-hmm. Right. You have, coming from this, you see all these convolutions? Mm-hmm. That's more realistic as compared to this, which only has a couple of twists and turns. But you see, look at that. That's a lot, isn't it? Because a lot of stuff has to happen here. And that would be proximal convoluted tubule. Descending limb, ascending limb, distal convoluted tubule.
The purple one.
In purple. By the way, you see how close this one is coming to the capsule? Remember, like we showed you here? Okay. And then into the collecting ducts, which they have painted yellow. Here, see what I'm doing here? My fingers here are what? The minor calyx is a minor calyx. By the time it gets to here, it is fully formed urine, because things are affecting the filtrate at every single step of the way, including the collecting ducts. All right? But as I said, I'll let your lecture instructor discuss that in much more detail. I'm less interested in that. Not that I'm not interested, but for our purposes over here. So we have this, so all of these. By the way, this would be more of a cortical nephron here, right? And this would be what? A juxtamedullary nephron over here. What are these? These things here. Now, remember we said if something is going to be reabsorbed, it has to be reabsorbed where? In the capillaries. In capillaries. Well, coming from the efferent arterioles, going to an interlobular vein. So from all of these, you have all these capillaries. They've only drawn one little teeny weeny region of capillaries. Because, you know, otherwise it would be one hell of a mess. And these are called the P-E-R-I, peritubular capillaries. Peri, like perimeter, around the tubular. And that's where everything is being reabsorbed into. Okay. Okay. Now, by the way, you see these vessels coming down here? These also have a name. Everything has a name. That's great for us. We can make tests. We have tests. We have to have the name. These are called the vasa, V-A-S-A, vasa recta, R-E-C-T-A. Are they in the book? They're in your sheets? Those are in the sheets too? Oh, son of a gun. Vasa recta. And these are the peritubular capillaries. Okay. What else do we have? Why don't you bring me that little one, that little square one with the black, somebody had one. It's in not great shape. In fact, it's in lousy shape. That's it, thank you.